Good morning. Welcome. Good morning and welcome everybody to the seventh lecture on immunology for students of Gaza. And we are now starting the second section of our lectures, which is going to be looking at innate immunity. What you're looking at here in this little inset picture here are some bacterial cells, and these cells have been engulfed by neutrophils. And all of this activity here is part of the innate immune system. This doesn't require antibodies. It doesn't require cytotoxic T lymphocytes to be recognizing a specific pathogen. Neutrophils are able to recognize broad classes of pathogens. Okay, so let's take this a little bit further, or let's take a little step back. Do you remember in our previous lecture, or a previous lecture, we looked at the role of macrophages and neutrophils, and natural killer cells, and the effective functions of natural killer cells are augmented, are enhanced by interferons and also these pathogen recognition receptors. The macrophages themselves are activated by pathogen recognition receptors after they recognize viral infection. Now, that's what we're going to talk about first. So our first subject in today's lecture of innate immunity will be looking at these pathogen recognition receptors. The pathogen recognition receptors are recognizing pathogen-associated molecular patterns, PAMPS. And these are general patterns, molecular patterns that are associated with a wide range of pathogens. And they're very general when we talk about general molecular patterns. Some of these recognition receptors are able to recognize microbial nucleic acids. And those are distinct in some ways from human cell nucleic acids. They're able to recognize lipids. They're able to recognize polysaccharides, and they're able to recognize proteins. So in other words, they're able to recognize each of the four classes of macromolecules that you'd study in any biochemistry course. So they can recognize a huge range of different patterns, molecular patterns within which are associated with so many different pathogens: viruses, bacteria, worms, parasites, fungi. And these were actually first identified or first recognized by mistake or by chance. The first pathogen recognition receptor was called the Toll receptor, and it was recognized by studies of Drosophila genetics. And this receptor, if you could look in a textbook of developmental biology, it'll talk in length about the role of the Toll receptor in setting up and controlling signaling gradients within the developing fly embryo. But as a side effect of studying this molecule or this gene, people noticed that if they knocked out this gene, which is a standard thing to do in molecular genetics type assays, then the flies are actually very susceptible to fungal infections. And what you're looking at here is actually a picture of a fly which has succumbed to a fungal infection. So without a properly functioning innate immune system, then we are very, very susceptible to a whole range of infections. And the Toll receptor was identified as a developmental molecule, and by chance its role in innate immunity was discovered later. And the Toll receptor is one of four major families of pathogen recognition receptors. When we say families, that means that there are many different members of those families. So there are many Toll receptors, and the family are called Toll-like receptors because Toll is one specific gene, but which was identified in flies, and genes which are homologous to that are many in the human genome, and they get the name of toll-like receptors. Pathogen-associated molecular patterns are indicated here in a general format, recognized by a pathogen recognition receptor. And the result of recognition of something foreign, something alien, not fully identified, maybe gram-negative bacteria, maybe a worm. It's not a very specific identification like that, which takes place between antibody and antigen. This stimulation of the cell results in the release of cytokines, and cytokines can interact with many other cells, immune cells like macrophages and dendritic cells. Phagocytic cells are a class of cells. So macrophages are phagocytic, but here is indicated primarily neutrophils. And they can result in cell migration, calling in cells from the bloodstream. Now, I call these cytokines, but as you can see here, these are called chemokines. And chemokines, those are simply a subset of cytokines, or cytokines whose function is known to be predominantly the migration of other cells in response to that signal. Then they're called chemokines. So chemokine is just a type of cytokine. We get cell differentiation. Remember that dendritic cells have these two forms. They have the highly phagocytic form, which after phagocytosis then the dendritic cell changes its form and is considered to be mature. It expresses epitopes, small fragments of the antigen that it's engulfed, that it's phagocytosed, and it expresses those complex with MHC on its surface. It stops being phagocytic and it becomes migratory and goes to the lymph node. You should recall from the last lecture that we saw dendritic cells in two places. We introduced for the first time the idea of the MALT, the mucosal associated lymphoid tissues. So first time we talked about dendritic cells, I explained about their migration from the tissue, carrying antigenic peptides to the lymph node. But in the mucosal surfaces, the dendritic cells are resident, and the cells that they want to signal to or they want to present to are also present there within diffusely defined tissues. So we have the encapsulated lymph nodes, just by way of reminder or revision, and we also have some of these areas of loosely defined lymphatic tissue, which is not encapsulated, where the dendritic cells and the CD4 positive T cells are located and interacting with each other directly in that same patch of tissue associated with the mucosa. Macrophages get activated. The endothelium is, as you know, it's a form of epithelium. It's not derived from hematopoietic stem cells, but more and more people are starting to think about the endothelium as, in addition to its epithelial-like properties of being a barrier tissue at cell surfaces, it also has such an important role in immunity. So it's an immune type tissue. The term pleiotropic or pleiotropic means that you get many different effects on many different cells. So the same cytokine may affect different cells differently depending upon what receptors are present on those cells to receive the cytokine, and also depending upon the activation status of various intracellular pathways between different cells. So the same cytokine or even the same receptor doesn't necessarily give the same effect. So this is what's meant by pleiotropic effects. So for those who have just now joined us, we've only just started, and we are looking at the concept of the pathogen-associated molecular patterns, which are recognized by families of pathogen recognition receptors. Now, something similar to pathogen-associated molecular patterns is the damage-associated molecular patterns. And it's a very similar concept, especially in the sense that there are specific receptors that recognize these patterns. So the same receptors, we call them pathogen recognition receptors. Uh, by the way, they may be soluble. That's a new concept I didn't introduce before. Before I showed them as associated with the plasma membrane. We will see a little bit later that in addition, these receptors can actually be intracellular as well. And that will be, the reason for that will be clear when we look at that. So regardless of where they are, these pathogen recognition receptors are able to recognize pathogen-associated molecular patterns. But they're also, and then give a cytokine response, which is going to stimulate the immune system, recruit phagocytic neutrophils, monocytes to the site of infection. In addition to that, when a cell becomes damaged or injured, all of those very unique contents of that cell will be released. And some of these, when I say some, actually many of them, serve as special signals which are recognized also by pathogen um, recognition receptors. And these are called damage or danger signals. So some D can stand for damage, D can stand for danger. Something's gone wrong in tissue if a whole load of intracellular molecules are suddenly being released. And the result of that is the initiation of an innate immune response. Normally, under controlled conditions of cell death, where cell death is actually regulated, then these intracellular components are not released, and the DAMPs are present, the danger-associated molecular patterns are present, or the damage-associated molecular patterns. But during apoptosis, the cellular contents are first of all degraded and also gathered together within granules, which then within that cell phagocytosed by macrophages. So there's no release of these danger signals. And the immune system then is quiescent. Quiescent sounds like it quiet, doesn't it? Quiescent means that that it's quiet. The immune system does not get activated with controlled cell death. But in the event of a severe injury, such as, for example, in the event of a burn, a severe burn, then we're going to have the release of many immunomodulatory danger signals. And you may know that we can define burns in terms of first or second or third degree burns. And the definition of this depends on whether damage is relatively superficial at the keratinized or only slightly at the epidermal layer, or whether damage penetrates through the epidermis into the dermis, or for third degree burns where damage has gone deep into the derma and even are starting to affect the subcutaneous tissue. And of course, we can have gradations between that. So the degree depends primarily upon the depth, but in assessing burns, there are also rules associated with the how much spread these burns are over the body. And these burns cause two problems in relation to immunology. The first problem is that all of the immune cells which are present here, and there are a huge number of immune cells present in the dermis and also in the epidermis, these cells are gone. So the physical barrier is gone. This thick protective keratinized layer is gone, and all the immune cells with this are gone, and the deeper tissue and the blood vasculature is exposed now to infection. And infection is a major corollary, major negative corollary of burns. And this can lead to septic shock and sepsis. But in addition to that, people have realized that even under sterile conditions, then there are very adverse effects associated with the overactivation of the immune system. And here's a recent paper which looked at the results of damage to the skin in the case of severe burns, damage associated molecular patterns, and the systemic, that means whole body wide, immune consequences of severe thermal injury. And they reviewed all of these molecules which are which have been considered to be danger associated or the damage associated molecular patterns. So some common ones, we looked at cytochrome C in the mitochondria. I think the first time a student looks at cytochrome C, it's in relation to the electron transport chain in the mitochondria and the production of ATP, or finally the production of ATP as a result of the activity of the electron transport chain pump or pushing protons into the intermembrane space, allowing for the generation or making a proton gradient, which is used to drive the ATP synthase. So that's the first time students generally see cytochrome C, but also the release of cytochrome C inside the cell is an essential is a mediator. I say essential because there is the mitochondria independent pathway. So cytochrome C is the mediator for apoptosis by the mitochondrial dependent pathway. However, once the cell itself gets disrupted and damaged, then cytochrome C is going to be released, and cytochrome C can then act as a damage signal. Mitochondrial DNA also is recognized as a signal. We saw earlier that mitochondrial DNA and as well as nuclear DNA can be released by granulocytes as part of a defense mechanism to trap various pathogens such as bacteria. And both of these can also serve as these danger signals. In addition, a whole range of other molecules, some of which probably are familiar to you, ATP, and some of which probably are not familiar to you. You don't need to remember the names of all of these. It's the concept that there are a wide range of intracellular molecules which, if released suddenly into the cell, are acting as a danger signal. And if these are serving as a danger signal, then there must be some receptors. And here is a list of various pathogen recognition receptors which are found on various cells: macrophages, neutrophils, endothelial cells. This is not an exhaustive list of cells, but these are cells where various of these receptors have been identified. I mentioned to you that there are 4 families, and here there is a number of different names. 2 of the major families are called the toll-like receptors, and I already mentioned that, as toll was the first pathogen recognition receptor that was identified, and later a whole class of receptors in humans have been identified. So this is one family, and NLR is another family. Presently, we will look at these 2 families and 4 other families. But before we do that, let's continue this issue of the severe burns and follow this pathway through. So far, we've gone from burns to release of danger signals, that there are cells which are able to recognize those signals. And what happens then is that signal is transduced to the nucleus, and that may be by a whole pathway, a whole series of steps that lead to the activation or translocation of transcription factors into the nucleus, which are going to change the transcriptional regulation. And these are 3 common transcription factors that have been identified as effectors Again, that word effectors that comes up a lot. Um, a final hand, if you want, that does something after some initial signal. So we get upregulation of many genes. Transcription factors generally don't function on one single gene, but they work together to stimulate uh, the expression or the repression sometimes of a whole collection of genes which work together in order to respond to whatever that signal was. And the response is we get the production of a number of cytokines. And this response can be harmful if the response is too strong, and we get what's called a cytokine storm. And cytokine storm is not uncommon uh, for severe acute respiratory distress syndrome. If you had a nasty case of SARS-CoV-19, then and you had and you were hospitalized, there's a chance that you would be suffering from cytokine storm, uh, and that can lead to multiple organ failure. And there's some of the target organs listed. And this is similar to what happens in septic shock, which is a very similar process. Instead of DAMPs, it's PAMPs, which are causing septic shock. And one of the most well-studied, if not the most well-studied example of a pathogen-associated molecular pattern, which is able to result in septic shock, multi-organ failure and death. This is lipopolysaccharide on gram-negative bacteria. And by the way, the LPS, which is a PAMP, also recognizes toll-like receptors. So these pathogen recognition receptors can recognize PAMPs and DAMPs. And the results are similar, which are activation of the immune system. For your information, it's worth bearing in mind that uh, there is a balance between activation and making quiet, and also there is research showing that uh, some of these patterns can actually uh, reduce some innate immune responses. So it's quite complex. You can have certain responses increased and other responses decreased. If we um, want to leave the question open, are we talking about activation, increasing the immune responses, or decreasing them? Then we can use the more general term immunomodulation. Immunomodulation. Listed here, for example, is interleukin 10, and that's an example uh, of a cytokine which actually uh, quietens down the immune system. There's one virus which infects humans, and it's transmitted to humans from animals, and that's the virus that I spent some time studying. It's called ORF virus. It's actually contains a mammalian homologue of interleukin 10 in its genome, and it's considered that this has been acquired by the virus by homologous or horizontal transfer, I should say. Sorry, horizontal transfer. So it's stolen there. It's stolen in the mammalian gene. And the logic, I suppose, is that this gene helps to make the virus invisible. Viruses have many mechanisms. We looked at one example when we looked at how vaccinia virus has some genes uh, which are preventing apoptosis, keep the cell alive so that it can still be a factor to produce viruses. So viruses also would like to make themselves invisible to the immune system. So production of interleukin 10 will keep the immune system quiet. So the concept here, immunomodulation, maybe activation or repression of the immune system. And even this term could be inaccurate in some ways. Perhaps we should say activation or repression of parts of the immune system, because many things function together. Okay, so one more thing to show you from this paper, which is that these actually have been considered to be promising targets for drug interventions. So we'd like to be able to block these effects in severe burn patients. And you don't need to memorize these again, but here is a list of various potential therapies um, which are targeting various known receptors or the specific danger-associated molecular pattern itself. And here are the cell types that are known to be involved. N is for neutrophil, T cell is obvious, M is for macrophage. And some of these can be applied topically and some of them by injection. And some of these have only been used in vitro. There are a number of studies being done in mice on these. The four families. I told you that there are four families of pattern recognition receptors. Here are the four families. We talked about this one a bit, and here is our friend the Drosophila who's lacking toll. And I mentioned this one, I haven't talked about it yet, and there are two others here. So there are some common patterns within the patterns, if you like. The first one is that two of these pattern recognition receptors are associated with membranes. So CLR, which stands for C-type lectin receptors, those are transmembrane proteins. And the toll-like receptor are also transmembrane proteins. And they can recognize a whole range of things. So bacteria, virus, that doesn't mean bacteria is only recognized by that and virus is only recognized by that. The picture is just showing you that there are a whole range of pathogens which can be recognized by these four family members. The unusual, interesting thing about the toll-like receptor is that it can also be found transmembrane, but in the endosomal membrane. So endosomes are a part of the what we call the endocytic pathway. When the cell wants to take something from the plasma membrane to inside, then it does that by endocytosis, often receptor-mediated endocytosis. And viruses, especially viruses, often find their way, and we've got bacteria as well in here, they find their way into cells via a receptor-mediated endocytosis. They're able to bind to something on the cell surface, and the cell is tricked into endocytosing those. And then those pathogens have a way to get themselves or their nucleic acid out of the endosome. So it makes sense then that being able to sense pathogen-associated molecular patterns inside the endosome would be very important, because that's a pathway, an important pathway by which many pathogens actually get into the cell. So we've got things actually at the barrier, on the plasma membrane itself, seeing what's around the side, but also inside these endosomes, then we have the ability to sense what's happening there. Okay, and the toll-like receptors are unusual and they have a very, very broad range of specificities. They can recognize proteins, nucleic acids, glycans, and other things. Glycans, what's a glycan? Glycan is basically a polysaccharide. And polysaccharides are extremely important targets for pathogen recognition receptors, because the bacterial cell wall is basically made up of peptides and sugars, oligosaccharides in a type of a cross-link. And those sugars in the bacterial cell wall are actually unique. They're different to those that are present in our cells. Even in terms of the stereochemistry, you probably studied before the D and L form of carbohydrates and also of amino acids, and our bodies utilize specifically the L form of amino acids and the D form of carbohydrates. But bacteria are not restricted to that, and so That's some aspect of distinction. So it's easy, relatively easy, to be able to distinguish pathogen sugars and the sugars present in our cells. Viruses also are generally, their cell surface proteins are glycosylated, and those can also serve as patterns for toll-like receptors. We'll see later how nucleic acids serve also as patterns. So very odd that the same family of molecules is able to recognize very different things. And the odd thing is that this family of molecules has quite a conserved structure. We won't really look at that structure much, but it's an unusual structure, and it's highly conserved, and yet can recognize so many different things. And of course, it recognizes those things, but doesn't recognize host cells. In this last bit here, it's talking about the pathways. The pathways. On the last slide, you saw this one here, NF-kappa beta. NF-kappa beta is a transcription factor. IRF is also a similar factor. These two factors come together. MAP kinase is an enzyme which is present in the cytosol. It's not a transcription factor, but its activation activates other enzymes which finally result in transcription. And it can also it can also signal through these. So we've got Shuhuwal. What is the receptor? What does it recognize, and how does it activate? And will you see this pattern here as well when we come to the C-type lectin receptors, transmembrane, as we said, but only on the plasma membrane. Recognizes glycans, so it's a bit more restrictive, and it recognizes glycans mostly from fungi, but also bacteria. By the way, it says fungi and bacteria, but there are examples of C-type lectin receptors that recognize viral glycans as well. Activation via a kinase called Psych and card9 malt BCL10 adapter complex. BCL10, you perhaps remember that BCL is a class of proteins very important in apoptosis, and this complex actually associates with the mitochondria. So mitochondria actually do many things, have many roles in addition to the role you study in introductory biology, which is in energy production. It's also involved in this innate immune signaling pathway. Now let's come to this one. NLR, it stands for nod-like receptor. You need to comprehend these four classes. The precise definitions aren't really something to remember too much, but NOD stands for nucleotide binding oligomerization domain. So N for nucleotide. So as the name indicates, it's recognizing DNA and recognizes viral and bacterial DNA. There are multiple subfamilies. So for each of the families, there are multiple examples. But with this family, it's actually very diverse, and there are subfamilies. And one of the subfamilies is called the NLPR. It's recognizing bacterial, viral, parasitic, and fungal pathogen-associated molecular patterns. And then this one, AIM2, which is recognizing viral and bacterial DNA. The interesting thing that these ones form multi-protein signaling complexes known as inflammasomes, and big bodies inside the cell that are able to activate caspase 1 and also able to activate various interleukins. When we say processing and activation, that's because interleukin 1b is made first in a pro form, and that has to be proteolytically cleaved. So that's processing. So processing, doing something physically to the pro form of interleukin, causes it to be active. And that's a common pattern that we see in biology of activation via proteolytic processing, and also interleukin 18. Inflammasomes are still a very hot topic in immunology and medicine, and they're being implicated in more and more um, immune pathologies, autoimmune disease, and hyperallergic responses. So, like everything in our body, they're doing something good for us, but they can become misregulated and can also be harmful, and they're targets for developing new drug therapies. This one is cytosolic and not associated, free floating. Is not associated with any membrane. So it's cytoplasmic sensor, and also this other class, the RIG1-like receptor. This is also a cytoplasmic sensor, and it's a sensor primarily of viral RNA. Viral RNA. So the R perhaps helps you here. Two R's, one for RNA, one for because it's a receptor. All of them end with an R because they're all receptors. RIG1-like. Sounds similar to toll-like, doesn't it? It's a common theme in molecular biology that at some point in history, somebody discovers something or understands the functioning of something, some gene, and they understand what that gene does, and then later they find that actually there are many other homologs. A homolog is something that at the nucleic acid or protein level is very, very similar, and is considered to be related. And this relationship may be very similar in terms of function, not necessarily, but generally that's the case. Okay, so this is again a family, RIG1-like receptors. So like toll in Drosophila is the archetypal, or the first case. There is something called RIG1, but this is in humans. But there are also a whole family. So this family is called the RIG1-like receptors. And RIG1 is very well studied, and that's an example of the RIG1-like receptors. Also, MDA5 is another very well studied member of that family. Okay. Now what we're doing now is going to look at some of these in a little bit more detail. And we'll start with the C-type lectin receptors. I wonder if you know what a lectin is. A lectin is a protein receptor that binds to sugars. That's what a lectin is. If I want to be more technical, I should say carbohydrates. But sugar sounds more friendly, doesn't it? Sounds more sweet. So here are some examples of different cell types, dendritic cells, monocytes and macrophages, and the Langerhans cell. We talked about Langerhans cells a few lectures back when we were trying to give a simple picture of the different cell types. And after giving this simple picture, I showed you that there's a little bit of complication in that actually defining cell types isn't as simple as it used to seem, because there are a whole range of intermediary cell types, and what appear to be very similar cells but actually have very different ontologies. Ontology means basically the family tree, or the ancestry, the way that this cell develops. So dendritic cells, for example, were initially or primarily thought to be derived from the myeloid progenitor cells. So your hematopoietic stem cell can finally become a dendritic cell via a myeloid precursor. Monocytes and macrophages also myeloid. But then we realized that actually some of these dendritic cells have a different origin, and some of them are actually lymphoid. The Langerhans cell is a cell in the skin, and some research papers call it a dendritic cell, and other research papers call it a macrophage. And it seems that when we look at cells present in the skin that are able to sense the presence of pathogen, there seems to be a whole spectrum of cell types distinguished by their different transcription factor, different transcription profiles. Sorry. The Langerhans cell is identified histologically and historically by the presence of langerin. And langerin is an example of a C-type lectin receptor. So in the skin, people identified a long time ago in histological sections a number of cells that they called Langerhans cells. And they called it this because of expression of langerin, and also because the name itself comes from a person who identified it. Afterwards, then people began to realize that actually these type of cells are present throughout the body, and they're actually macrophages. And then the complications started as people gained more knowledge of molecular markers to identify to question the true identity of the cell, and should it be a macrophage or a dendritic cell. Why is it called monocyte macrophage? It's because if you remember, and this is straightforward, this is from the first slide, macrophages don't circulate in our blood, but monocytes do. And monocytes infiltrate along with neutrophils after the initiation of an acute inflammatory reaction. So this is the beginning of the innate immune response. These cells, monocytes, get recruited along with the neutrophils. The neutrophils get recruited and do their job and die. The macrophages get recruited, the monocytes, and they turn into macrophages. And they seem to do their job, and some of them may stay in the tissue. Other macrophages actually began their life in the tissue in early embryonic stages and were not derived from monocytes, but from embryonic tissue, which we talked about previously. So these are examples. Dectin-2 stands for dendritic cell-associated C-type lectin-2, and this is the full name for it. Sometimes you see it called Dectin-2, sometimes you see it called CLEC6A. This slide shows you some of the accessory proteins inside the cell that actually are needed for signaling. So we've got the receptor. Up here it tells you what bacteria can be recognized by all these receptors. And some of them the same. The three cell types all have Dectin-2. As the name suggests, dendritic cell associated, which raises this odd question about, okay, here's a dendritic cell, but we find it here and we find it here as well. So receptors can recognize all these. And in order to send a signal, they need other proteins to be associated with the tail of the receptor. So most of the receptor is outside the cell. There's not enough amino acids inside generally to do very much. Sometimes there are, but often other proteins need to be associated in order to create these signaling complex and signaling pathway. And all of these are drug targets, for potential drug targets for medical purposes. Okay, if there are no questions about that. Okay, good. Here is an example of it's a very recent paper actually, looking at the role of one of these. And this is basically the same family as or same subfamily as Dectin-2, and it's a C-type lectin receptor. In this paper, the authors looked at a whole range of cancers, and this is an important area of medical application for immunology, is the relationship between tumors and the immune microenvironment. Many immune cells infiltrate the the tumor microenvironment, and they have important functions in trying to uh, control this tumor. Unfortunately, um, immune cells generally don't survive very long in the tumor, and they become tired, and uh, they lose their ability to control these tumors. So people are very, very interested in these cells from various angles, including trying to make them to be able to survive longer in order to control the spread of this tumor. In this particular case, the authors are looking at the correlation, and this is a common thing in molecular medical research papers, looking for correlations between gene expression of a particular gene of interest. So as we come along the scale, we have more and more gene expression. And the gene we're looking at is one of these C-type lectins called receptors, called CLEC4A. And looking for the correlation between that and something. And in this case, what's being looked at is within 17 different types of cancer, whether the outcome is favorable or unfavorable. In other words, how long the patient survived. So the authors took some cutoff and divided tumors divided into favorable and unfavorable. And also you took up the p-value, which is a statistical likelihood of the result occurring by chance. And usually it's the negative log of that p-value. It doesn't say negative here, but it is. And the colorectal cancer has actually, yes, I'm sorry, it's not, usually, usually such data are expressed as a negative log p-value, but in this case, it's not actually. It is actual p-value, but it's on a log scale. And so here is um, a tight cutoff, and 0.05 is the more relaxed type of a cutoff. The authors are only, out of all these 17 cancers, they're only interested in this one for their paper, because they go on to do studies in transgenic mice, which support the statistical association that in colorectal cancers, where the mean expression of this gene is upregulated compared to non-cancer cells, and that this is associated with a statistically significant favorable outcome. So this one experiment by itself would not be conclusive, but they begin to strengthen this result by, which I'm not showing you, but just to tell you, by doing some experimental work with transgenic mice. And they have a mouse model for colorectal cancer, and they can control the expression of this and see how quickly how long the mice survive with or without this gene expression. So innate immune signaling is important for not only in dealing with pathogens, but it's important in dealing with cancers. Oddly, in some cases, of course, I say of course, but I mean of course because you can see it here in the data. In some cases, actually, expression of this lectin is associated with unfavorable outcomes. So that's quite complex. People talk about cancer and curing cancer, but the question is which cancer? Each cancer has to be considered independently. There's no one, well, I'm sure you will know that, otherwise we'd have solved cancer a long time ago. There's no one treatment which works for all. Okay, let's move on to another type, the toll-like receptors. Remember that this is the one which recognizes the most diverse, the biggest diversity of pathogens. The toll-like receptors are numbered here, toll-like receptor 4, and toll-like receptor 4 is recognizing lipopolysaccharide. So I've talked so much about lipopolysaccharide. This is a gram-negative bacteria. In this case, it's flagellated. They're not all flagellated. By the way, flagella is, which is this tail which gives motility to some bacteria. It's very, very highly conserved in its amino acid sequence. Between different bacteria, there's very little variation, which makes it a very unique signal. And toll-like receptor 5 recognizes that. I'm coming back to the other side of the slide. LPS, lipopolysaccharide, which is a polysaccharide with a lipid tail. There's some diversity in LPS between bacteria, but toll-like receptor 4 is able to recognize them and initiate a response. But strangely, toll-like receptor 13 is able to recognize ribosomal RNA. So you may be aware that the ribosome, which translates messenger RNA, is composed of proteins, uh, dozens of unique proteins. It's an amazing machine. And in addition to proteins, there are some RNA molecules, ribosomal RNA. So we've got a toll-like receptor that can recognize those. There's also another toll-like receptor which can recognize DNA, in particular C's and G's. We've got toll-like receptors that recognize lipoproteins. So proteins can have lipids attached to them, and some of these can be distinctive of bacterial infections. And there are toll-like receptors that recognize them. The numbers may seem strange to you, the fact that here we've got 2 and then /1. What this indicates is that the structure of the toll-like receptor is actually a dimer. And it may be a homodimer, so 2 toll-like receptor 5s joined together, or it may be a heterodimer. And so here is these 2 are examples of heterodimers. So A was looking at bacteria. Toll-like receptors can detect viruses, and they can also detect parasites. Toll-like receptors can detect double-stranded RNA. Some viruses have double-stranded RNA. They can detect single-stranded RNA, ssRNA. Again, can recognize DNA with a slightly unusual composition. Again, lipoproteins, DNA with an unusual composition, single-stranded RNA, and profilin, and that's a kind of an indicator for parasites. Still with the toll-like receptor, here's the structure. I told you it's a dimer, and we mentioned that it's associated. It's a transmembrane receptor. So here's a membrane. The red atoms or the reds are indicating oxygens, and you can hardly see it, but there is actually some orange there, and that's the phosphate group. So you've got a phospholipid bilayer. Just to remind you of biology 101, perhaps. Yeah, this unusual shape of the molecule, like this big C shape here. And so two toll-like receptors actually have to dimerize. And now perhaps when you look in the middle, you can see what this is. What is this, do you think? This red strand, and then there's a dark blue strand. This is double-stranded DNA. And without going into much detail, but this protein actually has a whole selection of leucine residues, amino acid residues called leucines, which form this arc-type shape. And in the picture, it kind of makes sense that it's shaped in a way that can bind to DNA. However, it's not as simple as that in reality, because the same shaped molecule can recognize a huge number of other things. So that's one strange aspect of that. I think we might leave it to the next lecture to introduce the idea of how the toll-like receptors signal. Okay, I'll introduce the idea. I think that's good. Pathogen-associated molecular pattern, pathogen recognition receptor. Toll-like receptors use this pathway to signal through. This pathway is common actually to many signaling pathways, so you will see it again in other places, maybe in other courses, not necessarily mine. NF-kappa beta is a very common transcription factor, and it can be stimulated by more than one thing. Amongst them, these toll-like receptors. What's listed up here, remember I told you that commonly cell receptors need to associate with many other proteins in order to transmit their signal. And the toll-like receptor is no exception. And importantly, this signal activates something called the IKK complex. And what this one does is it results in the separation of an inhibitory molecule. So here in these red circles, this is the transcription factor. It's a dimer. And this NF-kappa beta is just waiting to be free and to go inside the nucleus and do some gene transcription. But it can't do because this molecule, and the I stands for inhibition, this molecule is preventing it from going into the nucleus. But as a result of the toll-like receptor binding to a pathogen-associated molecular pattern, that receptor gets activated and sets up a signaling pathway, which results in the phosphorylation of this molecule, and that phosphorylation sets up the inhibitory unit to go and be degraded by the proteasome. Good. So next time we have a lecture, I'll take up this story again, and we'll also look at the other pathogen recognition receptors. Okay, thanks very much for your attention, and see you next time.